Good afternoon, everyone.
Good afternoon.
Thank you very much, Dr. Vanderpue, for such a nice introduction. So, as Dr. Vanderpue said, I am Dr. Chaudhary. I am from Albany State University, working in Center for Nanobiotechnology Research, which is an independent research center but associated with Albany State University. So today I'm going to talk about the nanomaterials and their antibacterial activity and their peptide conjugates and how they can be used as effective antimicrobials in order to treat different bacterial infections. So I'm going to present a part of my research here today. In the center, we work on various nanomaterials, and mostly we investigate the biomedical application of these nanomaterials. These nanomaterials are already characterized, and we buy it from company, and what we see is how they can be used for the treatment of different diseases or different conditions. So to start with my lecture, I would talk about the research themes that we carry out at our center. So there are three major aspects that we cover in the center. First one is we evaluate the anti-RSV. RSV is a virus, a respiratory syncytial virus, which affects infants and the older ones, wherein the immune system is compromised, right? They are more prone to infections. So in the center, what we are trying to do is we are trying to develop the vaccine against this RSV infection by incorporating the nanomaterials and their biomedical applications. So that is one field. Another field is use of nanomaterials as antibacterial agents and investigating their nanogenomic, proteomic, and physiological studies on bacterial organisms. And the third field is how the nanomaterials regulate immune response in the host cell. And we study incorporating nanoimmunology and nanogenomics. So I'm going to talk about the use of nanomaterials as antimicrobial agents in my presentation. So these are some of the important bacteria that we study in our center. These are all biosafety level 2 bacteria. As you are already aware of different biosafety levels of different organisms, BSL-2 organisms are basically the ones which are relatively safer compared to BSL-3 and BSL-4, meaning that under normal microbiology, standard microbiology conditions, you're free to work with them, ideally. So these are some of the important BSL-2 pathogens which causes several infections in human beings as well as in animals. So as you know, like bacteria are classified depending upon their staining properties and cell wall. So we work on 2 grams positive strep and staph, which are mainly involved in skin infections. They cause cellulitis, and this cellulitis can really go very bad if not treated properly. And mostly gram negatives, which are foodborne also, like Salmonella typhimurium, E. coli, and Pseudomonas. Pseudomonas is basically, it causes cystic fibrosis in the patients. It's a nosocomial infection. You can say the infections that originate in the hospitals. And E. coli, as you know, 0157 strain is a very dangerous strain that causes colitis, hemorrhagic colitis, a lot of blood loss in feces, and you kind of become anemic, and eventually it leads to death. So these pathogens are very important as far as the human life is concerned. So we try to target these pathogens in order to see if nanomaterials are effective against these pathogens to treat them, and if they are, then what is the underlying mechanism? We try to define that too. So as you can see, there are 2 phenotypes of this particular bacteria, Pseudomonas, a mucoid and non-mucoid strain. The mucoid strain is more resistant to any antibiotics, and these are the 2 strains that we study in the lab. So as you can see, some of the nasty lesions induced by these pathogens, cellulitis, then bacterial keratitis, enteritis, and ear infections, which if you are not treating on time, they can really cause serious damage. So as far as the trends in development of new antimicrobials, from 1983, it has gradually reduced, and the main reason for that is development of antibiotic resistance. This is a very, very common problem nowadays in order to treat the bacterial infections. And the reason is, number one, due to excessive use of antibiotics. I mean, there was no limit of use of antibiotics, which resulted into acquiring the resistance pattern among the various bacterial pathogens, and they became resistant to different commercially used antibiotics. And also, due to the lack of knowledge, if sometimes the choice of antibiotic is not being used and some wrong antibiotic is being used, then the bacterial species tend to acquire the resistant genes, and then that's how they develop the resistance against particular bacterial pathogens. So as you can see that the antibiotic resistance started evolving, and within a very short period of time, like within 10 years, sometimes within 3 years, or maybe over a period of time too. So it's gradually increasing against most of these commonly used antibiotics. And therefore, we need to come up with a strategy that can prevent antibiotic resistance problem. And the best solution for that is use of these carbon materials. They are basically graphene rings, and there are different carbon materials like nanotubes, then carbon blacks, carbon fibers, and most of them, they have basically applications in battery operations or water purification systems. Especially these single-walled nanotubes are used for water purification systems in order to reduce the bacterial load in the water. And the biggest advantage of using nanomaterials is their size. So they are so small that they may go unnoticed by the human body immune system, and they reach to the specific target, especially the intracellular pathogens like salmonella or other intracellular pathogens they can target. So with that thing in mind, people have started using metal nanoparticles. It is very common. It is very commonly known that several metal nanoparticles are very good antibacterial property, especially gold, selenium, and they have several biomedical applications. They can be used as anti-cancer, then drug delivery, then antiviral. And in CNBR, we mostly work on silver nanoparticles, silver-coated carbon nanotubes, silver PVP coated silver, gold and spions, that is superoxide ion super magnetic peroxide ion oxide nanoparticles, and then functionalized nanoparticles. Why we use that, I'll cover it in my next slide. So today I'll just talk about the silver coated CNT, the antibacterial property of silver coated CNT and their peptide conjugate. So I'll just cover this topic in my presentation. So as you can see that this is a typical silver coated CNT. This is a carbon nanotube, and these black deposits are silver deposits on it. This is a TM image of it. This work has already been published. So my supervisor, he was working in collaboration with another university, and they kind of developed a method where they can deposit silver on carbon nanotubes. So it's a kind of functionalization. So it's a coating of carbon nanotube surface with silver nanoparticles. Why silver nanoparticles? Because silver nanoparticle has antibacterial activity, and also CNTs have an antibacterial property. But silver nanoparticles tend to agglomerate. They kind of form a precipitate in a solution. And CNT provides a good host material where it can lie on. So that's why the combination of these two is much more efficient compared to only silver nanoparticles and CNTs. So this was the work that they published using some gram-negative and gram-positive pathogens, and they were able to show that compared to, this is a Kirby-Bauer assay, and compared to only silver, the silver CNT-treated bacteria had more zone of inhibition. So in order to evaluate the antibacterial activity of the silver-coated carbon nanotubes, this is what we do in the lab. We first investigate their minimum inhibitory concentrations. That is the concentration that is needed to kill the 50% of the population. Then we do live dead viability staining, growth curve analysis by exposing the bacteria to various concentrations of silver CNT, and evaluating the survival in a time-dependent manner, like 0, 4, 8, 16. Then we do electron microscopy in order to capture the morphological changes induced by these nanomaterials in bacteria. And then we perform molecular studies like what is happening at gene regulation level, and also check protein profile. So this minimum inhibitory concentration assay is based on the resazurin reduction dye-based method, wherein the color of the dye is reduced from violet to pink with the enzyme reductase produced by the viable cells. Meaning that when you add this dye into a solution wherein the bacterial cells are present, that dye color will be reduced from violet to pink, and that's how you can estimate the growing population. So as you can see clearly in this slide, this is a this is a 96 microliter plate, which contains different dilutions of nanomaterial, starting from 500 microgram per ml and all the way to 0.9 microgram per ml. Then there's positive. Positive means actually there is no treatment with nanomaterials, and negative means there is nothing, absolutely no bacteria, no nanomaterial. So as you can see, the color of the dye remains violet at higher concentrations, meaning that it's killing all the bacteria added to the plate. This is after 24 hours, and since there is a pink color, the dye is reduced. That means there is a growing population in this one. So somewhere the MIC lies somewhere between 7.8 to 3.9. So you can define the MIC level in this one somewhere between 7.8 microgram to 3.9 microgram per ml. And this is the peptide concentration. So yeah, this is what I was talking about. So these are silver CNTs concentration from 0 to 125 microgram per ml. And as you can see, the MIC lies between 31.2 to 15.6. I want you to remember this concentration for silver CNT. It is somewhere between 31 to 15. Why? I will explain that as I'll go along with my presentation. So CNTs are toxic to, they have MICs between 31 to 15 microgram per ml. So then we go do live and dead bacterial staining. There are Cyto9 and PI dye, which stains the cell green when they are viable, and if the cells are dead, they'll stain them as red. This is basically PI staining that gives the red color, and that's how you can differentiate between the live and dead population in the treated and non-treated samples. So this is control, E. coli control, which has not been treated, and this is E. coli treated with silver CNT. So as you can see, the number of dead cells are more in the treated samples. Then we do growth curve analysis by simply exposing the bacteria to 4 different concentrations, which lies within the range of MIC, starting from 50 to 0, and do the plate counting assay, calculate the actual bacterial number over a period of time, and just to quantitate, this is a quantitative analysis, that how many bacteria are surviving at particular concentration after a particular time. So, and then we perform electronic microscopy, scanning electron microscopy and TEM in order to capture the changes. So as you can see, these are healthy bacteria. And if you compare it with the silver C. enties one, you can see some dissolved kind of entities here, damaged bacteria, lysed bacteria. Also, there is a hole inside and the bacteria is kind of melted down. And upon TM, as you can see, the nanomaterials actually enter inside the bacterial cell wall, break down them, and then causing lysis, and also creating some empty ghost-like structures. Then we carried out qPCR in order to check what is happening at gene regulation level. We picked some of the important genes which are involved in ABC transporter genes and amino acid biosynthesis, which show downregulation of these genes. This ABC transporter genes system is present on the membrane of bacterial cell wall, which allows transport of different proteins across the cell membrane. So since the membrane was damaged in treated group, the gene regulation was also found to be downregulated. And same was the case in case of amino acid biosynthesis related genes. Then the DNA transcriptional activator were also downregulated. There was no significant difference for FLEH, but SDI and YBF. These particular two factors are responsible for quorum sensing mechanism of bacteria, which is very important. It's kind of communication way between the bacterial species and their host. That process is called as quorum sensing. Nowadays, it is a very hot topic of research because bacteria or pathogens kind of communicate with their host in order to protect themselves. So if they see any danger, they kind of generate molecules around them and gives a signal to the surrounding population that something is going wrong, so you need to adapt. And that's how they protect their community inside the host. So this SDIA gene regulates the expression of that receptor, which actually catches the quorum sensing molecule. So that was also downregulated. And YBEF is a biofilm formation related gene. As you know, like it is another way of bacteria to protect themselves, to form biofilms on a surface. So what they do is they extra polysaccharide layer is being formed around the microbial communities so that the antibacterial agents wouldn't be able to penetrate through and kill the bacteria. So it's a kind of umbrella they hold and they hide, behind the umbrella. So the antibacterials would not be able to reach and kill them. Then some of the virulence associated, what is virulence, right? These are the factors which are required by bacteria in order to produce disease. So those factors were also, some of them were downregulated and some of them were upregulated. This upregulation could be a Phoenix effect, that you just treat them and with a particular concentration and it just boost up. So, but most of them were, they showed downregulation, which indicate that it has something to do with the virulence associated machinery too. So as I discussed, like some of the genes were upregulated, mostly SPI2 related, and then mostly the outer membrane associated and ABC transporter genes, the genes that maintain cytoskeleton of the bacteria, those were downregulated. And we also did mass spectrometry. This was not done by me. There was a previous researcher who did this, but this was very interesting, so I thought I'll produce this result. So as you can see, there were 4 samples, non-treated, only carbon nanotube treated, silver coated carbon nanotube treated, and with the 2 different concentration. And with this combination, bless you, with these 2 combinations, the expression of those proteins were reduced drastically. The protein expression was found to be reduced. And these are just bunch of genes, but there were like a lot of other genes which were found to be, whose expression was reduced compared to untreated one. But the main problem with this silver coated carbon nanotube is its toxicity to our normal human cells. You remember I said the MIC is between 31 to 15. I told you to remember that because anything in between that range is toxic to our body. So if we take that, it's not good for our healthy normal cells. Then what's the use of using silver-coated carbon nanotubes, right? What is the use of using it? But that is still being used in the topical application. Like if we have skin infections, those are not recovering, silver nanoparticles are still used on, they are still used in those creams in order to treat the topical infections. But in order to make it more efficient, what is the strategy? Like there are a lot of different strategies. So nanomaterials are very good source of delivery systems for various cancer reagents. So that they lead to targeted drug delivery, and hence they have been shown to be very effective in order to treat the cancers. I mean, at least in vitro analysis have shown that. So we tried that, like there are several antimicrobial peptides which could be either commercially prepared or host originated. These are very small amino acid sequences which are prepared synthetically or which are derived from the host, which holds antimicrobial property. This is commercially available, and our center has collaboration with the company, so they provide us these molecules and we kind of test them. So in order to avoid the in order to address the toxicity issues of the silver-coated CNTs, we decided the conjugation of these peptides with silver-coated carbon nanotubes. So these antimicrobial peptides are very effective. In our center, we evaluated this, and the results were quite impressive. This is 10 to the 9 CFU per ml, meaning that very high number of bacteria were used in order to evaluate this. But if you reduce the number, which is standard, as per the standard procedures, which is 10 to 5, the concentration is very low that is required to kill the bacteria, which is 0.3 microgram per ml for some of the peptides. So the point is they are very, very effective. But the major problem with that is, this is the mucoid strain, mucoid and non-mucoid strain of Pseudomonas aeruginosa. The growth curve, like they were exposed to different concentrations of peptide and evaluated for their survival by plate counting method and also optical density method. And then this is Salmonella typhimurium exposed to one of the peptides. As you can see, it was very effective. Almost all the concentration, no bacterial growth was observed. And if you see, the concentration is pretty low, like 8 microgram per ml, even 1. And then this is the live date staining. The similar results were observed, like maximum bacterial cells were found to be dead. This is for Salmonella typhimurium and E. coli. And as you can see, that peptide and peptides, this AP antimicrobial peptide had different antimicrobial activity. From the ACM, we could see that they actually create a hole. If you can see this centrally placed, sometimes they were placed on the periphery of the bacteria. They kind of form a hole and try to take out all the cytoplasmic contents and make the bacteria as empty ghost cells. Of course, this was the sub-inhibitory concentration that was used to capture these morphological changes. At MIC, that bactericidal concentration, everything is dead. So this could be the possible mechanism. From these results, we interpreted that it is kind of coming in contact with bacteria and then creating a hole and then causing expulsion of all cytoplasmic content and eventually death. Some of these pictures show the live cells. You can see. Then the TM analysis showed, if you can see, the membranes are broken down, and these are the 2 different strains of Pseudomonas aeruginosa. Empty ghost cells are being formed in the treated one. See, the membrane is damaged. And also it kind of regulated the gene expression that is responsible for mucoid to the transformation of non-mucoid to mucoid phenotype in Pseudomonas aeruginosa. So these results were indicative that even APs, antimicrobial peptides, are also very effective. But these results are all like in vitro analysis, like done on it's like bench work outside. The major problem with these peptides is they have sensitivity issues. So if they are very sensitive to pH changes, so if the pH of the solution change, the activity may differ. They may not be active. So that being said, and the toxicity issue with silver CNT, why not to conjugate these two components and see what is happening? That was the idea, and that's what we are doing currently. So as you see in the genes, yeah, this is what I was talking about, cytotoxicity assay. So at this particular concentration, if you see the silver CNTs were toxic, like almost 30% cells, almost 70% cells were dead after the treatment over 24 and 48 hour time period. So this all showed, and these are 3 different cell lines, HEP2, J774, that is murine macrophage, and A549, which show that silver CNTs within their antimicrobial concentrations are toxic to the normal human body cells or normal cells.
So they're all human cell lines, right?
What?
They're all human cell
Yeah, these are murine.
Oh, murine.
Yeah, and these are human cell lines. Yeah. This is lung carcinoma and this is hepatocellular carcinoma cell line. But at the same time, the peptides were not toxic. So they were still safe to use for the human use. But as I said, the pH was the major issue. The different conditions, if they change, they tend to lose the activity. That was the threat. So that's why we came up with the approach like, why not to conjugate them with the nanomaterials in order to come up with a effective antimicrobial strategy. So in this word, the process is called as bioconjugation, wherein the biological molecule is attached to a nanomaterial by the process of chemical bonding. It could be either covalent or non-covalent. Yeah.
I missed the first part of it. How does the silver bind to the carbon nanotube?
It was chemical deposition method.
So it's inside or outside?
It is on the surface, outside. Yeah. So, and that is commercially available. So we bought it from company, and they use CVD method for preparation. Normally, what they say is they tend to create the carboxyl groups on the surface of the CNTs, and those carboxyl groups are filled with silver CNT. That was one of the explanation provided by the company person.
It's about how many silver atoms per unit length of carbon nanotube?
Yes. I don't know, like, they defined how many actually molecules are there per carbon nanotube. But they kind of define the size of carbon nanotube, which is from 1 to 5 nanometer, and the length is 1 micrometer. So as you can see that this is silver CNT and attached to the functional group, then it can be stronger combination. So this is the simple thing that we did. We mixed both the peptide and silver CNTs together, then keep it for shaking 30 minutes, then added, the first silver CNTs were treated with EDC and NHS. These are 2 cross linkers. They kind of create carboxyl group. So the attempt was to conjugate them using functional groups. So EDC and NHS would activate the carboxyl groups present on the CNTs theoretically, and peptide has amine group. So with the combination of this EDC and NHS, there will be a strong covalent bonding between peptide and silver CNTs. And then further, we characterized them and also investigated their antibacterial activity. So this is a very popular method. That's what they do, like carboxyl containing nanomaterials. The carboxyl groups are activated using EDC and NHS chemistry. Maybe a chemist person could define this in a better way, but this is what they use in the paper. And once the carboxyl groups are activated, it forms a bond between carboxyl group and amine group, and that's how it's conjugated. Then after that, we characterize them using zeta potential and size. Zeta potential showed that there was a shift in charge only silver CNTs had minus 30 MV as a charge, but after the peptide functionalization, the charge was shifted towards positive side. This may indicate that shift towards positive side indicate that the agglomeration property is more, which was true because they were not easily mixable, and carbon nanotubes are not soluble in any of the solutions. But nowadays the companies are coming up with soluble silver CNT solutions too. So this was a kind of confirmation that the functionalization has happened since there was an increase in size. Of course, the size is just a rough estimate because this determines the dynamic diameter, which is not real, which is not true because they are cylindrical in shape CNTs. So these values tend to differ, but the shift in charge clearly indicated the functionalization occurred. And then we carried out FTIR, Fourier transmission infrared resonance analysis, which shows the presence of functional groups in a given molecule. And as you can see that peptide, the mixture had the similar peaks as that of peptide, and some of the stretches were similar in the functionalized silver CNTs, same as that of peptide. And then we investigated the antibacterial activity, and as you can see, the MIC value was reduced somewhere. First it was somewhere between 31 to 15.6, but it was reduced one-fold in case of E. coli and Salmonella typhimurium. It was twofold. So that definitely, functionalization definitely helped to reduce the minimum inhibitory concentration of silver CNTs, meaning that if there was a symbiotic relation between the peptide and silver CNTs in order to produce better antibacterial effect. However, we are now characterizing more about how the proper way of functionalization can be achieved, because that was just a simply mixing method, which we never know that how much conjugation is occurred. So to functionalize them with different peptides and try to come up with a strategy which one is the best, which combination is the best, and also characterize them using different techniques. Also, we'll be investigating the effect of functionalized silver CNTs in using animal and in vitro models in order to see whether the toxicity is reduced or if the toxicity is less compared to only individual components of each of those. So I'd like to acknowledge NSF and NIH for their fundings, all the students, postdocs working in our center, faculty who are directly or indirectly related to our center. Then these are the three main pillars of our center. Dr. Singh, who's the director, is a professor of microbiology. He is the main person who kind of brings all the funds and funding source and is supporting hand for all of us. Then Dr. Sri Kumar Pillai, he's a professor of biology. He's also one of the co-PI in the CREST. And Dr. Dennis, she's also one of the co-PI in CREST. Then this is our wonderful staff that is always being supporting for us. Like, she's the lady who handles all the instruments, and these are our staff which make us feel like a home at ASU. And this is our group, all the students and professors. I hope you enjoyed my presentation. Thank you.